This is lecture 3 from the chapter 1 on viruses. We left off, we had gone over the first two groups of the Baltimore classification. And just as a reminder, Baltimore classification is based on the type of nucleic acid that is in the core of the virus, and then the mode of messenger RNA production. So basically, each one of these groups, if we go back to the central dogma that we learned in Bio 111, how proteins are produced. Sorry, that's just supposed to say protein. But anyway, if we go back to the central dogma, group 1 has a double-stranded DNA molecule and produces messenger RNA in the traditional way that you see written up, under the title. Group 2 has a single-stranded DNA, which simply converts to double-stranded DNA and then completes what, the same process that group 1 completes. So the single-stranded DNA simply replicates a complementary copy of itself. And in group 3, the double-stranded RNA molecule is used to transcribe a messenger RNA, and then the process, the messenger RNA is used to produce the protein. So it's a double-stranded RNA, and the double-stranded RNA takes the place of the DNA in this step, and it produces a strand of messenger RNA, and then the messenger RNA codes for the protein. All right, then there is single-stranded RNA positive and single-stranded RNA negative. They react differently. So if it's single-stranded RNA plus, that's group 4, the genome actually functions as RNA and skips transcription. Okay, so absolutely skips transcription. An example of a single-stranded RNA positive virus is the common cold, or picornavirus. I actually think that first R is not supposed to be there, but I have to look it up. I think it's just picornavirus. But anyway, regardless, it's the virus that causes the common cold. And notice it's an RNA virus, and RNA viruses are more likely to mutate. They have a faster rate of mutation. And we can't make a vaccine for the common cold for that reason. Now, the 5th group is single-stranded RNA negative. And in that case, messenger RNA transcription does occur. Messenger RNA is transcribed from the RNA genome. So single-stranded RNA transcribes messenger RNA, and then messenger RNA codes for protein. And rabies is an example of a single-stranded RNA negative virus. Then we have group 6, which are single-stranded RNA viruses with reverse transcriptase. We call these retroviruses. Group 6 and 7 are both retroviruses. Okay, so both of these groups, group 6 and 7, are retroviruses. Group 6 and 7. But in group 6, you start out with a single-stranded RNA virus that contains its own reverse transcriptase. So it's going to start with RNA, use reverse transcriptase to make DNA, and then it will complete transcription and translation in the normal way. In group 7, and by the way, HIV, human immunodeficiency virus, the one that causes AIDS, is an example of this type of virus, group 6. And group 7, an example would be hepatitis B. So that's a double-stranded DNA virus with reverse transcriptase. So how does this work? The viral genome is double-stranded DNA, but the viral DNA is replicated through an RNA intermediate. The RNA may serve directly as messenger RNA or as a template to make RNA. So this is a very complex process. The viral DNA is replicated through an RNA intermediate and then basically works like groups 4 group 4 or group 5. That messenger RNA that's produced, either transcription is skipped, or that messenger RNA is used as a template to make that RNA intermediate is used as a template to make messenger RNA. So it either works like RNA positive or RNA negative viruses. So yes, this is a little complicated, but you can always refer back to this Baltimore classification when you're taking your quiz. You know, you can just pull it up or pull your notes up, pull your slides up, and have them available to you when you're taking your quiz, you know, if you need it. So, okay, now the impact of viral infection. So what happens when a virus does infect an organism? It says the viral replication cycle can produce dramatic biochemical and structural changes in the host, and that may cause cell damage. And these effects are called cytopathic. Path refers to disease and cyto refers to cell. So cytopathic means they are effects that can cause damage to a cell. So these cytopathic effects can change cell functions or even destroy the cell. The symptoms of viral diseases result from the immune response to the virus, which attempts to control and eliminate the virus from the body and from cell damage caused by the virus cell. The virus, I think that means virus host cell. The virus itself, in other words. A virus is not a cell. So I'm sorry, it's just a bit confusing the way they wrote it. All right, and then lysis, budding, and apoptosis. Lysis means breakdown. Budding is the process where reproduction, where an organism is produced as a little bud off of the adult, and then it pinches off and grows into its own adult. And apoptosis is programmed cell death. It's when cells actually destroy themselves. They're programmed to do that in certain conditions. Some infected cells, such as those infected by the common cold virus, which is a picornavirus, but it specifically is known as rhinovirus, rhino meaning nose, actually die through lysis, bursting, or apoptosis, which is programmed cell death or cell suicide. So the common cold just kills your respiratory cells, the cells in your respiratory tract. And that's what's going to cause all your symptoms that you get, you know, because it's cells that are lining your nasal cavity and your you know, your windpipe and your bronchial tubes and those kinds of things. That's going to be affected. So those cells are dying, and that is the reason for the symptoms that you get. And also the inflammation that results from your immune system trying to kill the cells that are infected with the virus. So every time a host cell does die or burst, it releases all the virions that the host cell produced at once, and those virions can go on and attack new host cells. Many animal viruses, such as HIV, leave the infected cells by a process known as budding. So basically you have, here's your host cell. In HIV, the host cell is actually a T cell, which is one of your white blood cells. And if you destroy T cells, you basically shut down the immune system, which is what we know the AIDS does. So, so let's say this is the HIV virus. Sorry. It basically, budding in this case is like exocytosis. So basically, here's your HIV virus, and then it goes like this. Then the plasma membrane sort of surrounds the HIV virus, and eventually it pinches off. Okay, so some viruses leave by a process known as budding. And in that case, the virions leave the cell individually and they have an envelope around them. During the budding process, the cell does not undergo lysis and is not immediately killed. However, the damage to the cells that the virus infects may make it impossible for the cells to function normally, even though the cells remain alive for a period of time. Sometimes the host cell lives on and continues to replicate the virus. So the viruses that are released from cells by budding just have a different mode of causing symptoms. So steps of viral infections. We actually learned first about viral, you can't really call it a life cycle, but this is kind of like a life cycle. So the cycle of a virus, from one virus to making many viruses. We actually learned about a bacteriophage first, so that's why we're going to use this example. Other viruses behave similarly, but not always the same way. But anyway, so what you see here is the bacteriophage, which has a complex shape. It's a naked virus, but it has a head and a tail structure. So the first stage is that the bacteriophage attaches, it's called attachment. It attaches to the host cell. And what you're looking at here is the bacterium is a host cell, and we know the DNA of a bacterium is a circular chromosome. So that pink structure there is its circular chromosome. All right, so the virus attaches and then it injects its DNA into the host cell. That's called entry. And the DNA incorporates, the bacteriophage actually replicates the viral DNA. And then the viral DNA is used to make the proteins that surround the DNA. So basically, you have replication of the viral DNA, that's replication stage. Then in the assembly stage, the viral DNA codes for the components of the virus. And the components of the virus are assembled. So basically, the components are simply the capsid and the viral DNA. So the DNA is enclosed inside the head portion of the capsid. And now all these viruses cause the cell to lyse, and that releases the new viruses, the new virions that are produced. And they can go infect another cell. Now, there are two cycles with bacteriophage. There's the lysogenic cycle and the lytic cycle. We just learned the lytic cycle, okay? So now let's talk about the lysogenic cycle. In the lytic cycle, the viral DNA did not incorporate into the bacterial chromosome. But in the lysogenic cycle, notice that the phage DNA or viral DNA becomes incorporated into the host genome because the viral DNA is green and the genome of the bacterium is purple. And then when the cell divides, every time it divides or reproduces, it reproduces by asexual reproduction or cloning. It's called binary fission. Each new bacterial cell that's produced is going to have a copy of that viral DNA, which under stressful conditions, so let's say that it's a herpes virus, a person who has contracted the herpes virus has that condition for life. But if they have a lot of stress in their life, whether it's physical or mental, any type of stress can cause them to have an outbreak. And when that happens under stressful conditions, the viral DNA separates itself from the bacterial chromosome and it looks like a plasmid actually, but it becomes its own DNA and enters the lytic cycle at this stage, at the replication stage.